Hi, I'm Apollonia Gardner, and I'm a junior in computer science, biology, and neuroscience, and today I'm going to tell you about my project on the striatum. So there are three things you need to know. The striatum is a brain structure linked to Huntington's disease. It contains two types of cells, striosomes and matrix cells, and it's directly involved in cost-benefit decision-making. And so the goals of my project are to characterize the roles of these two cell types in decision-making, and also to run wild-type and Huntington's disease mice through these behavioral studies to figure out if there's an underlying circuitry change in Huntington's mice. And so I'll first talk about my calcium imaging results. So calcium imaging in general is a way that we can see cells lighting up in the brain and correlate that activity with behavior. And so here I'm showing you 4 different types of T-maze tasks that we run our mice in, and each of them are characterized by different levels of cost and rewards on either side, the rewards being different levels of chocolate milk and the costs being different intensities of light. And below I'm showing you heat map plots of neural activity for striosomes and matrix neurons in the 4 task types, where red represents high activity and blue represents low activity. And so, for example, striosomes in the benefit tasks seem to be relatively active regardless of whether the mouse goes towards the pure chocolate milk side or the mixture. Whereas in the cost, striosomes seem to be relatively active when the mouse goes towards the high cost side but not the low cost side. Behaviorally, we see a striking change between Huntington's and wild-type mice. So Huntington's disease mice seem to be relatively repetitive and fixed in their behavior. So given their choice on a previous trial, they tend to make that same choice on like the next trial, 2 trials after, 3 trials after, 4 trials after, versus wild-type mice, which their choices are largely unaffected by the history. And lastly, we use electrophysiology to kind of visualize the spike waveforms that we're recording over here. So here I'm showing you 2 different classes of neurons in the striatum, and they have different spike waveforms. This one goes up and then down, this one goes down and then up. So we already know that there are different types of neurons in the striatum and that these belong to different groups. And then based on the time between spikes, this neuron has very small times between spikes, so it fires really fast and doesn't stop. This one has more variable time between spikes, so it doesn't fire as reliably. And so based on these properties, we can classify neurons into different types. And so for future directions, we're going to run more mice through this pipeline to kind of resolve some of these behavioral and neural activity trends better to see if we can elucidate Huntington's disease.